Hello students, now we will discuss about mitotic cell division. Mitotic cell division is also called as equational cell division or indirect cell division. And in case of mitosis, one mother cell gives rise to two baby cells in which the chromosomal number that is present in mother, it is equal in case of two babies. It is not divided into half. So mitosis, also called equational division, indirect division. So in case of mitosis, two daughter cells are obtained from a single mother cell. Here mother cell means the cell which is giving rise to two daughter cells, mother cell, and maintaining same number of chromosomes like that of mother. So mitosis, where does it takes place? Mitosis takes place, or mitotic division takes place, in somatic cells. Somatic cells are the cells except the reproductive cells in any type of living organism. Mitosis in plants is explained by Strasburger, and in animals it is explained by Flemings. So the ploidy that is indicated for mitotic cells is diploid. The ploidy used to indicate mitotic cells that we call as diploid. Next, what are stages that are included in mitotic division? So if a question arises in reproductive cells or in germinal cells, mitotic doesn't take place, answer will be there will be one exception. That is, mitotic division in germinal cells, it takes place during multiplication phase only. So mitosis in germinal cells or germ cells takes place in multiplication phase. Next, so what are the stages that are included under mitotic division? Stages included are: first one, karyokinesis. Second one, cytokinesis. Means after mitotic division, cytokinesis happens. Karyo means nucleus. Kinesis means division. Division of nucleus is called karyokinesis. So again it is of 4 different types of stages. It is of 4 types. What are those? One is prophase. Second one, metaphase. Third one, anaphase. Fourth one, telophase. After interphase, when cell is ready for mitotic division or getting divided into 2 baby cells or 2 daughter cells, then what happens? So we will write after interphase. During interphase also, the cell will be having all the cell organelles, nuclear membranes, etc. Just we will write one small diagrammatic representation regarding that. So think that these are chromatin material, like this, which are covered by nuclear membrane. And there will be cell organelles and cytoplasm here. So this is cell. These are nuclear membrane. Here we can differentiate like what type of mitosis. Mitosis is it same in case of plants and animals. There is one small difference. There are some special type of structures called as aster rays, which are not seen in plant cells but seen in animal cells. So we can write here mitosis in animals. In animal cell. So, nuclear membrane, this is cytoplasm with cell organelles. So, this is chromatin. Chromatin means now the total genetic material is not differentiated into chromosomes separately, but all together it is combined like a highly coiled genetic material. Later when it divides into pieces, then it will be called as chromosomes. Chromatin. Chromatin means highly coiled chromosomal material or genetic material. In a long thread like form. So after this happens, after means when the cell prepares completely itself with all the requirements, then it starts prophase. What happens during prophase? This prophase in case of mitosis, it is divided into 2 types, early prophase and a late prophase. Early prophase, late prophase. In case of early prophase, what is happening? This total highly coiled chromosomal material or chromatin, now it will get means condensed. Now we are telling this chromatin now is in decondensed form. In case of early prophase, this one starts getting into condensed form of chromatin. It means that now the chromatin material is ready to get converted into chromosomes. So in case of early prophase, so we'll write some of the points. All cell organelles starts disappearing. Even nuclear membrane also disappears. Then how the structure is looking at early prophase. So now the nuclear membrane starts to get disappear. The centrioles that are present on the means exactly now they are not moved to opposite poles. You can write somewhere here like this. So these are centrioles. This is disappearing nuclear membrane. And even cell organelles also got disappeared. Now here chromatin condenses into chromosomes. So now here by early phase it means that chromosomes are formed. If you write one complete structure of a chromosome, it will be looking like, we can say this is X chromosome. This one is called arm. This is called arm. This is called arm. This is called arm. The central one is called centromere. Centromere on either side is having some special structures called kinetochore. Spindle fibers during mitosis will be attached to this kinetochore only. Two vertical arms, this upper arm and lower arm, together are called chromatid. So here in this chromosome, we can see two chromatids. I think now you got what are labelings of a chromosome. If you take X chromosome as an example, this one is called one arm, second arm, third arm, fourth arm. Two vertical arms are called one chromatid and middle one is called centromere. On either side will be present kinetochore. Late prophase. In case of late prophase, so here we have written about centrioles. This is the cell. Here will be centrioles which are moved to opposite poles. Since we are discussing about mitosis in animal cell, we can say if it is plant cell means we will not draw the extra structures like aster rays. What are aster rays? Aster rays will hold centrioles at opposite poles. So there will be some gathering of aster rays. These are also made up of special type of tubulin proteins, etc. So we can say these are aster rays. Since we are discussing about mitosis in animal cell, we are drawing aster rays. If it is plant cell mitosis means we will not draw aster rays. Next, as we have said that in early prophase, nuclear membrane got disappeared and chromatin material has got condensed into chromosomes. Now, we will draw chromosomes here. Along with these chromosomes, there will be formation of a spindle fibers also. Spindle fibers are made up of tubulin proteins. Like this. So these are spindle fibers. Since here spindle fibers are starting from one centriole to other centriole, these are called as continuous fibers. Continuous fibers. Now, even at late prophase, here the kinetochore will be there on either side of the centromere that has to get attached to one centriole here. So these spindle fibers which are now formed, we will change the direction of these chromosomes. Two chromosomes we will write. So this will be kinetochore, this will be kinetochore. The spindle fibers will be formed newly and gets attached one end at kinetochore, other end at centrioles. Now, when the spindle fibers are touching to either side centrioles, those are called as continuous fibers. Now, what about the centrioles that are connecting? only one side of the kinetochore. These are called discontinuous spindle fibers. Discontinuous spindle fibers. Here, spindle fibers are made of tubulin protein. Two types of spindle fibers are formed now. And since we can see these aster rays are 2 in number formed at opposite centrioles, so that we can say the condition is amphiastral. 2 aster rays formed at opposite centrioles are said to be in amphiastral condition. Means having 2 aster rays at opposite centrioles. What is function of these aster rays? They hold centrioles at opposite ends. Function is holding centrioles at poles. So this is about interphase, means before starting of M or mitotic division. Prophase, early prophase, these are the points. You can draw just this diagram roughly. And late prophase. Now in the late prophase we are seeing formation of aster rays, spindle fibers, and that too here in late prophase we are seeing 2 types of spindle fibers. One is continuous spindle fibers and second one is discontinuous spindle fibers. Here there are some other terms for continuous and discontinuous spindle fibers. We will write that terms also. Continuous spindle fibers are called interpole fibers or interpolar fibers. Discontinuous spindle fibers, also called as chromosomal spindle fibers or chromosomal spindle fibers. So when a question has come like how many types of spindle fibers are there, answer will be according to the type of stage that cell division is existing in. In late prophase there will be 2 types, continuous and discontinuous, which are also termed as interpolar and chromosomal spindle fibers. After late prophase, the next one will be metaphase. So in a cell, after the formation of spindle fibers, 2 types of spindle fibers, what is happening at metaphase? These chromosomes will be aligned on equatorial plane like this, on same line in the middle. Still they are attached. This will be the centrioles. These chromosomes are attached to either side of centrioles like this, to the kinetochore like this. But all the chromosomes will be on same line. We can also give aster rays and also other type of spindle fibers which are continuous like this. So this will be the cell. These are continuous spindle fibers. This will be chromosomes on equatorial plane. These will be aster rays. And this will be discontinuous spindle fibers. We can write some of the points during metaphase. Here, during metaphase, the exact shape, or we also can say morphology of the chromosome or structure of the chromosome, is well studied in metaphase. Morphology or structure or and shapes of chromosomes. Shape of chromosome is well studied in metaphase. Actually, this is a previous year MCQ. You can say this is MCQ. So here in case of metaphase, we can take one point like these are all aligned on one equatorial plane. On one equatorial plane. Next. And these spindle fibers are still attached to the kinetochores here. There are different, we have written here the shapes of chromosomes. What are different shapes of chromosomes? Different shapes of chromosomes are K-shaped, L-shaped, and J-shaped and I-shaped. That we will see later. But first we will finish Anaphase. So just I will write K-shaped, J, L-shaped, and I-shaped. I will draw the diagrams of those shapes later. First we will see about Anaphase. So third one, Anaphase. In case of Anaphase, the chromosomes that are aligned on the equatorial plane, they will be moved to or dragged to opposite poles by spindle fibers. But before that, what happens? Chromosomes are splitted at centromeres. So we get chromatids getting separated. First we'll draw the diagram. Centrioles, centrioles. Asteroids. Next, continuous spindle fibers will be as it is, there is no change. But there will be change in the size of discontinuous spindle fibers. So, first step is chromosomes are splitted at centrioles. Sorry, at centromeres. We will write that point. Chromosomes are splitted at centromere. Okay. Next. So that what happens? These spindle fibers which are attached to the kinetochores, they will drag concerned chromatids towards opposite poles. So here in the middle part, other type of spindle fibers are formed. These are called as interzonal spindle fibers. These are called interzonal spindle fibers. These continuous spindle fibers are also termed as interpolar spindle fibers. And discontinuous spindle fibers are called as chromosomal spindle fibers. We'll write some points regarding Anaphase. Chromosomes are splitted at centromere. At centromere. We will remove this point also. Next, spindle fibers drag chromatids towards opposite poles. Now we can see in Anaphase only we can see interzonal spindle fibers. Now, one more point. For dragging of one chromosome towards opposite poles, approximately more than 30 number or approximately 30 number of chromosomes, so ATPs are required. So where to write that point? 30 ATPs are required to drag one chromatid to opposite poles. Now, after Anaphase, the next stage will be Telophase. So here we have seen in early prophase, the cell organelles, nuclear membrane, everything is getting disappeared. The thing will be reversed in case of telophase. Here, all cell organelles and nuclear membrane, I am writing as capital N, nuclear membrane reappears. When we go for diagrammatic representation, so here, okay, the chromatids that are present in Anaphase, now they start to decondense into chromatin. So here, chromatids are there, they start to undergo. So when this upper part of chromatids have been decondensed, nuclear membrane will be formed around them. The lower part of chromatids, the nuclear membrane will be formed around them. Chromatids decondense to chromatin. At the end of telophase, means here we are talking about animal cell, so there will be invagination at the cytoplasm, like this. Spindle fibers, aster rays, everything will be dissociated. Now here I did not represent any of the spindle fibers or aster rays, means they got disappeared now. Cell organelles will start forming now. Now next stage will be cytokinesis. Since we are talking about animal cell, the type of cytokinesis is centripetal type of cytokinesis. Type is centripetal. It means the invagination or the division of the cell into two daughter cells that starts at the ends and will be processed towards the central region, like this. Means division of cytoplasm starts at edges, or we can say at the borders, and it will be penetrated towards the center from borders to center. Center of cell, so that in the next stage we will get two baby cells. So we can say two daughter cells have been obtained. Now there will be chromatin and also the cell organelles, and again chromatin and also the cell organelles along with cytoplasm. We can say these are daughter cells. Actually, to represent the total types, 4 different types of stages: prophase, metaphase, so metaphase, anaphase and telophase, I have given along with diagrammatic representation. Even for board writing students or NEET students, regarding the mitotic division, almost all points have been covered.